Hi, everybody. So today's lecture will be lecture number 6, Biochemical Methods. So this will be the only lecture in the entire course where we'll discuss specifically some techniques or methods that you would use in a biochemical laboratory to investigate specifically proteins. So we'll start out with a discussion on how you would purify proteins, so an individual protein away from other proteins in a cellular mixture, and how you can analyze how pure your protein is. Then we'll move on to talk about how antibodies can be used as biochemical tools to identify specific proteins. Then we'll talk about a few different techniques that you can use to actually determine the primary sequence of a protein that you've purified. And lastly, we'll talk about two methods very briefly on how to determine the three-dimensional structure of a protein that you've purified. So I hope you all enjoyed the lecture, lecture number 6, Biochemical Methods.
All right, so we'll start off today with a quick discussion on what the proteome is. So you've probably heard the term the genome. So the genome is the entirety of the genetic material that's found in an organism's cell. So the human genome has about 3 billion DNA bases, and that encodes about 23,000 different proteins. So the number and type of genes expressed into proteins is different for each cell, and that changes with the physiological state of the cell. So the state of protein expression of a cell or a collection of cells at a given time is called the proteome. So it's an important concept to remember that the genome is fixed. So your genes can't change. The DNA encoded in your genome is fixed. It's not changeable. But the proteome is dynamic. It's going to change over time and be different between different cells. So some dynamic features of the proteome are, what proteins are expressed at a given time, what's the abundance of each protein relative to other proteins, so the relative gene expression, and how does protein expression change under specific environmental conditions. So, on top of this, biochemists also want to know what function each protein performs and how these proteins perform that specific function. So, an important first step for this, which we're going to go over today, is to purify a specific protein away from all the other proteins in the cell, and then you can study its behavior outside the cell or in vitro, as opposed to in vivo, which would be studying that protein's function within the cell. So the first step in purifying a protein or getting it away from all the other material and other proteins within the cell is we use differential centrifugation. So cells are first disrupted or lysed, so you break open the cells using something called a homogenizer. So you would use this to break up the cells and get all the material, all the liquid in there, homogeneous. So that's why it's called a homogenizer. So the outer membrane is broken, which releases the mixed intracellular components. So then we can then sequentially centrifuge that homogenate by increasing centrifugal force. So you can see up here at the top, we do our homogenization. So we form a homogenate. Then we centrifuge that homogenate and at a sort of low speed. So in this example, it would be 500 times g, so 500 gravity forces for 10 minutes. And so that would then separate the nuclear fraction in the cell away from the cytosolic fraction. So the nuclear fraction would be in this pellet here. And so we would keep that in the tube, and then we would take out the remaining liquid on top, and that would be called the supernatant. So the supernatant is going to contain everything in the cell except for the nuclear fraction. So you can keep doing additional rounds of centrifugation, and at each step, denser material will form a pellet, and less dense material will remain in the supernatant. So these isolated fractions, which is what they're called, they can be used for further purification of your protein of interest. So let's say your protein's located in the nucleus. Then you would take that pelleted fraction and actually purify it from that. Let's say your protein's not located in the nuclear fraction, then you would take the supernatant and purify the protein from that, and so on and so forth. And there's very defined parameters as to how much you need to centrifuge a specific sample in order to get a specific pellet or a fraction of the cellular material. Now, before you would even purify your protein, you need a way to make sure that your protein is actually in the fraction that you're interested in. So to do this, you develop what's called an assay or an in vitro assay. So this is a specific biochemical test that can be used to confirm a specific protein's presence based on some functional aspect of the protein. So, for example, if we wanted to purify the enzyme lactate dehydrogenase, we would take our purified fractions that we think contain lactate dehydrogenase. We can then add lactate to this solution and NAD plus, and then the activity of lactate dehydrogenase is to convert lactate into pyruvate and to reduce NAD plus to NADH. And NADH can actually be measured using light absorbance at 340 nanomolars wavelength. So for any different protein that you're interested in identifying, you first need to develop an assay so that you can, actually confirm that the protein's in the particular fraction that you've purified. So now once you know that your protein is in a fraction, there's still going to be a lot of other different proteins in there, and you really want to purify your protein to a degree to where you can study its activity, maybe its structure, and stuff like that. So there's a few different ways that you can further purify your protein using chromatographic methods, and one of the ways that's commonly used is called gel filtration chromatography, and this is a size separation way of purifying different proteins or separating different proteins from each other based on their size. So, in this method, you basically take a column that's filled with these beads that are sort of porous. They're usually polyacrylamide beads. And so they have they're sort of porous gel beads. Now, larger proteins are going to be less capable of entering these beads, so they're going to go through the column faster. And so when you apply pressure and time and let those proteins separate out, you can then collect these different fractions containing proteins of different sizes. And you can know that there's a protein there based on measuring the wavelength, the absorbance at a wavelength where proteins will absorb. So as you can see here, so it's gel filtration chromatography. It's a method used to separate proteins based on size. Larger proteins are going to go through the column quicker, and smaller proteins are going to get stuck in those beads for longer and then elute out uh, much slower. So you can separate the proteins by size using gel filtration chromatography. So another way that you can separate proteins from each other is using ion exchange chromatography, and this is going to be a method to separate proteins based on their charges. So all different proteins have different amino acid compositions, and so proteins are often going to have an overall positive or negative charge, and you can actually just type in the sequence of your protein, and there's a pretty good chance that that there's computer programs that can estimate the overall charge of your protein. So what you would do then is take a column and that contains beads with the opposite charge to what your protein's overall charge is going to be. So in this example here, we have negatively charged beads, and then so your positively charged protein is going to bind to those beads and sort of be stuck on that column. And then other negatively charged beads are going to be repelled by the beads and basically just flow through the column. So now you've separated out positively charged and negatively charged proteins. You can also do this with positively charged beads if your protein has an overall negative charge. But in this example, now these positively charged proteins are going to be bound to these beads. And then you can take a salt gradient, so just slowly add more and more salt, and that'll compete with the protein for the binding on those beads. And so eventually that's the way you can actually remove your protein from that column. So ion exchange chromatography, another way to purify proteins, is very commonly used, and it's going to separate proteins based on their overall charge. Now, another really common way of separating your protein from other proteins is using affinity chromatography. And this actually just separates proteins based on some specific function that the protein has. So, the proteins will be isolated based on their affinity for a particular model. So you just a molecule, sorry. So you just take that molecule and attach it to a bead and then run your mixture of proteins over it, and then your protein's going to bind to the bead, but other proteins that don't have that particular affinity are just going to flow straight through the column. So the example that they use here is if you wanted to study a glucose binding protein, you can take beads that are covalently attached to glucose molecules, and then run your mixture, your homogenate, your fraction over this column, and then your protein, because it binds to glucose, is going to bind to these beads, whereas other proteins that don't have glucose binding ability are going to flow right through. So the protein can then, once it's stuck to the column, you can release it by just adding free glucose, and then that's going to compete with the beads for binding on your particular protein. Now, there's a lot of different affinity chromatography methods and columns that are available and that people use based on the particular features or the particular affinity of your protein for that bead. This is just one example, but there are many, many other ones, and you can separate a lot of different proteins very easily using this method. So affinity chromatography, which is based on the function, it's a separation based on the function or a particular quality that your protein of interest has. So now we've gone over some different methods used to purify proteins. But once you have a fraction and you think your protein is in it, you need a way to actually determine that your particular protein is present. And one of the first steps that we would use to determine that is to use what's called an SDS polyacrylamide gel electrophoresis, or SDS page for short. So the first step in this technique is to add the detergent sodium dodecyl sulfate, which is also called SDS for short, and you add this to a mixture of proteins that you're wanting to analyze. So SDS does two important things. So I want you to remember these two things. So number one, it denatures all the proteins. So the proteins are now not folded into their particular conformation. And number two, it overcomes that inherent protein charge. So we just talked about that proteins could have a charge on the outside of their protein. Well, when you add SDS, it both denatures the protein and it sticks to the outside, giving all of them a negative charge. So now all the proteins are denatured and have a negative charge. So what this allows you to do is to separate the proteins by size alone. That's the only characteristic that you're going to be separating proteins by when you use an SDS page gel. So you now can take that protein, put it in a gel, and then run a current in it that goes from negative at the top to positive charged at the bottom. So you run that current, and all those negatively charged proteins are going to move through this polyacrylamide gel. And now larger molecules are going to move slower through this polyacrylamide gel, and smaller proteins are going to run faster. So this is going to allow you to analyze what proteins are in your particular fraction or your particular, after a particular purification step, to see what proteins are still present. And if you know the size of your protein, it can help you figure out if your protein is actually present in that fraction. So SDS page, very important method for analyzing proteins, especially when you're doing purification methods. And so here's a particular picture, a visualization of an SDS page gel. So you have that gel, you ran your protein mixture through it, it's separated by the proteins by size, and now you can visualize the different proteins in your gel using Kumasi blue staining. So Kumasi is just a chemical mixture. You add the gel to it for some period of time. You actually heat it up a little bit, and that makes it go a little quicker. And this chemical will turn the proteins in that SDS page gel blue. So you can see, just like we talked about, larger proteins are going to be closer to the top of the gel, and then smaller proteins are going to be closer to the bottom. And you can actually buy what are called ladders, so just mixtures of different proteins of known sizes, and then you can say, oh, my protein is about this size, and I know this protein is about this size, so I'm thinking that my protein is in this particular lane, and it's purified to this degree. So you can see there's a lot less mixture of proteins in this lane as opposed to this lane. So this is how you would visualize protein separation on an SDS page gel. And so if you're running multiple different rounds of purification to get your protein of interest as pure as possible, you can use these SGS page gels to look at the relative purity of your proteins compared to everything else. So this is just sort of a cartoon example. So you have your homogenate here, which contains a lot of different proteins. Salt fractionation we didn't talk about, but it's sort of a weaker way of purifying proteins from other proteins. Ion exchange chromatography, so now you separated proteins by charge. Let's say your protein was positively charged. This would be only the proteins that have a positive charge. You got rid of the protein with the negative charge. Then you used molecular exclusion chromatography, so you just purified proteins that are about this size here using that method. And then lastly, let's say you used affinity chromatography to purify your specific protein based on some function. Now you have a really pure fraction of your protein of interest, and you can, save that and run all sorts of different assays to study its activity. So this is sort of just bringing together how you would use SDS page gels to measure the different levels of purification when you're purifying a protein. So I'm not going to talk too much about this, but you can also use these polyacrylamide gels to separate proteins on other features rather than just size. So one of these is isoelectric focusing. So in this case, we talked about how proteins have different charges on the outside of the proteins. You can actually separate them by the relative strength of their charge using isoelectric focusing, which you basically make a page gel with a pH gradient. So they'll migrate to their isoelectric point, or their PI, which is the point where the protein has a net zero charge, and then it's not going to migrate any more, or a net neutral charge, sorry. And one note with isoelectric focusing, so SDS cannot be used in isoelectric focusing because the proteins have to be charged. And of course, SDS both denatures and gives all the proteins a homogeneous negative charge across the sample. So if you really wanted to separate proteins out by both size and charge, you could actually do two-dimensional electrophoresis. And so this would be the combination of using isoelectric focusing, separating proteins by their charge. And so you would do that first, and then combine that with this SDS page gel to separate proteins based on overall size. So two-dimensional electrophoresis really allows you to separate proteins out, and it would give you a sort of gel that looked like this, where the proteins aren't just in a line in one lane, they're actually spread out over 2 dimensions, so hence two-dimensional electrophoresis. And those 2 dimensions, of course, are overall charge of the protein and the size of the protein. So now that we've talked about some basic protein purification and analysis methods, we'll move on to talk about antibodies as biochemical tools. So antibodies are very important and widely used in biochemistry labs. So antibodies are immune proteins that bind to specific regions called epitopes on specific proteins called antigens. So you've probably all heard about antibodies before, especially in recent years. So if your body is invaded by a microorganism, the microorganism is going to have proteins on the outside. Let's say it's a virus, it's going to have proteins on the outside of the virion. Your body's going to recognize those, produce antibodies that bind very specifically to that protein region, which would be called the epitope on that protein, which would be referred to as the antigen. So biochemists take advantage of this and use antibodies to identify specific proteins, which allows us to, analyze their activity and all sorts of different stuff, and to see whether they're present in a sample. So there's two different types of antibodies that we would use in the lab. So one are called polyclonal antibodies. So these are a collection of antibodies of different that bind to multiple epitopes on a single antigen. So let's say there was a protein that you're interested in. Well, these antibodies aren't going to bind to the whole protein. They're going to bind to specific motifs or regions on that protein. And those specific small regions are called epitopes. So if you have polyclonal antibodies, you have a bunch of different antibodies that are going to bind all to the same antigen, but to multiple epitopes on that specific antigen. Monoclonal antibodies are a bit more specific. So they only bind to a single epitope on a single antigen. So they're very, very specific for your protein. And there may be different reasons why you'd want to use polyclonal antibodies versus monoclonal antibodies. So there's different benefits and drawbacks to each. And then monoclonal antibodies actually used as a therapy in disease. But we don't need to, that's sort of above what we're going to talk about today. So today we're just thinking of antibodies as biochemical tools. So going back to the purification steps that we were talking about before, antibodies can actually be used to purify specific proteins. So like we said, their antibodies are very specific for specific regions on specific proteins. They're only going to bind to that epitope and hopefully not epitopes on other different proteins. So you can use this to do a technique called immunoprecipitation, where you're going to take your homogenate mixture, add your antibody to it, and then isolate that antibody that's bound to that protein. And then you can later release that protein from it. So immunoprecipitation is a really good way to isolate your specific protein. And you can use this for protein purification methods, but also just to, if you want to look at your protein of interest, this is a really good way to do it. It can also be, let's say you were looking at a protein-protein interaction in a cell. You say, oh, I think this protein binds to this other protein. You can use immunoprecipitation to, quote unquote, pull out your protein of interest, and then you can see if that protein that it's binding to is actually present in that sample as well. So that would be called a co-immunoprecipitation. So immunoprecipitation, a very important tool that uses antibodies for purification of a specific protein from a mixture. So antibodies can also be used just to detect whether a protein of interest is present in a biological sample. And so this technique is called an ELISA, or an enzyme-linked immunosorbent assay. So this is a very common diagnostic tool also. So if there's probably many different biological tests or medical tests that you've gotten that use an ELISA assay. So pregnancy tests are basically just ELISA assays. And so ELISAs are most useful when proteins are present in large quantities in the sample. So they're not incredibly sensitive, but they can be sort of used. So they use antibodies combined with a colorimetric assay to determine whether a protein is present in a sample or not. So there's two different types of ELISAs. We can go through those quickly. So there's an indirect ELISA, where you would basically take your sample, you'd coat all the proteins in that sample in a well, in like a plastic well, basically. And then you take a specific antibody that you know binds to the antigen that you're looking for in the sample. You then wash it, and then you link that to another antibody that has a specific enzyme attached to it. So if that antibody binds to your antibody, then it'll change color, and you can say, oh, yes, this protein is present in this sample, or no, this protein isn't present in this sample. A sandwich ELISA is a little different, so that you would just take your antibody to the antigen that you're looking for, and you'd actually coat it with the antibody. Then you'd add the sample or the antigen and then wash it. If it binds, then you would add another enzyme or antibody that has an enzyme linked to it, and if it changes color, then your protein is present. So ELISA, it's a very important tool, and I don't expect you to remember all the different steps or indirect versus sandwich ELISA, but know that it uses antibodies to detect a specific protein in a sample using a colorimetric assay. So now next we'll talk about Western blots. So Western blots are also another way to use antibodies for detection, but it's very useful if there's only a small amount of a specific protein in a sample. So what you would do is you could run an SDS page gel using a specific, that you think your protein is present in at a certain concentration, maybe, and then you would take this gel and just using an a voltage, you would transfer that to a polymer sheet, and then you would incubate that with the antibody to your protein of interest. So in this gel, you're going to have a whole mixture of different proteins. But when you mix it with the antibody should just bind to your specific protein. So then you can hit it with a specific wavelength of light, and then that causes the protein band to be detected by fluorescence emission. So the antibody bound to your specific protein on the gel, and then that makes a fluorescent line on the immunoblot, you would call it. So Western blots are very important to detect small amounts of proteins. Also, if you have two samples that you treated differently and you think, oh, well, if we treat it this way, then the concentration of my protein is going to go down, you can actually run those side by side and use Western blots as a quantitative assay, or a semi-quantitative assay, to determine that, oh, yes, the protein concentration is going down because there's less protein now present in this. So Western blots combine SDS page with antibody detection, and they are very sensitive and useful to detect a very small amount of protein in our particular sample. All right, so now that we've gone through how you would purify a protein and how you would detect that a specific protein is present, now we can talk about, well, what if there's certain information that you want to know about that protein, including maybe its primary sequence. So let's say it's a protein that you're seeing in gels, but you have no idea what the sequence is. Well, there's a few different methods that you can use to determine its sequence. So the classic example is Edmond degradation, and so this is a way to determine the protein sequence of a polypeptide that you've purified. And so this is used, how this works, and I've actually never done this method. So the next one I'll talk about is a bit more common right now to identify individual to determine the actual sequence of your protein. But Edmond degradation is sort of the classic way to do this. So you would you would have your protein, which has a specific sequence, and then you would add this reactive compound, so phenyl isothiocyanate, and then it would actually couples and labels the end of the protein, so the N terminus of the protein, and then you can release that molecule off. And so you're basically shortening the chain by one amino acid each time. And then you would use some other method to determine, which amino acid was removed at this specific step. So it's one by one determining the sequence of your protein. And this is called Edmond degradation, and it's more of a classic way of determining protein sequence. So the much more common method and much more sensitive method and the easier method that most people use now to determine a protein sequence is called mass spectrometry. And so mass spectrometry can be used to identify proteins or peptide fragments derived from protease digestion, but it's used to identify proteins based on their mass. So if you let's say you ran an page gel and this one particular protein band keeps coming up, you can actually take that protein band and run it on mass spec, and that would allow you to determine its sequence and maybe its identity, and then maybe it's doing something pretty important in your sample. So there's a lot of different types of mass spectrometry, but the important thing to remember, just like the name says, is that it identifies proteins or separates proteins based on their mass. So the example below is a time of flight mass spectrometer. And this basically, you're shooting proteins through a tube, and they're going to move uh, differently. The speed at which they move through that tube is going to be different based on their mass. And so, this is a very, um, very sensitive way to separate proteins based on their mass and to get very, very accurate uh, determinations of the mass of a specific protein or a peptide fragment derived from a protein. So, and then you can run this through a lot of different computer programs, and they can sort of help based on databases of particular protein sequences and particular amino acid sequences. You can link the mass that you determined of your peptide fragment by mass spectrometry to a specific amino acid sequence. So this is a very sensitive and very useful method for determining protein sequence, and it's a mass spectrometry is also used for all sorts of other different studies, and it can be very important for studying things like the proteome. So you can take a sample if you want to see, in this cell at this particular time under this particular treatment, what kind of proteins are being expressed compared to other ones. Mass spectrometry can really help you determine what proteins are present in that sample because it's going to separate them by size, and it's going to do that very accurately. So, mass spectrometry, a very sort of modern way to separate proteins based on their sequence. And so I mentioned it a little bit before, but mass spectrometry can also be used with a protease degradation or some sort of degradation step to determine a protein sequence. So you combine protein degradation step with mass spectrometry. So you can use a protease digestion, or you can actually use that Edman degradation method and basically just cleave off individual amino acids one at a time, and then you can run those different fragments through the mass spec, and this actually allows you, using computer programs or something, to determine the mass to charge ratio, which gives you a very accurate determination of the protein's specific mass. So tandem mass spectrometry, it's called that because you're using a degradation step and a mass spectrometry step in tandem. And so this is a really good way to determine a protein, primary sequence of the protein. And so now we've gone through protein purification, different ways to identify what your specific protein is, if it's present in the sample using antibody methods. We've talked about, once you have your protein, how you can determine its specific primary sequence or, primary structure of that protein. But what about if you want to determine the actual three-dimensional structure of your protein of interest? So I'm not going to go through big detail on this, but if you want to determine the three-dimensional structure of a protein, there's basically two methods that are available to you. So one is X-ray crystallography, and the other is nuclear magnetic resonance, or protein NMR. So these can both be used to determine the three-dimensional structure of proteins. So X-ray crystallography, that's more bio background. I used to do a lot of X-ray crystallography research. And so how X-ray crystallography works in a very general sense is you would take your protein, purify it to a very high degree, and then you can crystallize it, and then you basically mix it with different chemicals, which allows you to make a protein crystal. And so this crystal can then be shot with X-rays, very high quality X-rays, usually at a synchrotron. And then the way that the X-rays bounce off the specific atoms in that crystal allows you to determine the three-dimensional structure of the protein. Now, this has been a very important method in biochemistry. So this is the method that they use to determine the structure of the DNA double helix. All of the proteins that you've seen so far have either been found out, determined mostly between X-ray crystallography and protein NMR. There are a few other methods that can be used to determine three-dimensional structure, but these are the two most commonly used ones. So X-ray crystallography, you purify your protein, you then try and crystallize it, which is, not a trivial feat. It takes some time, and sometimes it doesn't even work. But if you can get good quality crystals, hit them with X-rays, then you can actually determine the structure of the protein. And how you determine it is because that information gives you you can determine where in three-dimensional space that electrons are present. So that would be this blue sort of mesh. That means that there's electron density at that position, and then you build your protein model within that electron density to determine the three-dimensional structure of the protein. So this was a picture here of a protein bound to a carbohydrate, and you can see the carbohydrate molecule, even the orientation of the carbohydrate molecule, and then a phosphate molecule here in the active site. So then another method that you can use to determine the three-dimensional structure of proteins is using protein NMR. And so with protein NMR, I'm in no way an expert on protein NMR, but as far as I understand, one of the benefits of it is that you don't have to crystallize your protein. You can just purify your protein, have it in solution, and then you basically hit that with very high-powered magnets that actually align the nuclei of the proteins, of the atoms in the protein in your sample. And that gives you an understanding of the interactions between the different atoms in your sample, and you can use that to determine the three-dimensional structure of your protein using NMR. There are some size limitations to this, so this doesn't work very well on insanely large proteins, but it does work well on a lot of different proteins, and it also provides you more information on the dynamics of the protein, so how proteins moving in three-dimensional spaces and how it maybe interacts with other proteins. So with X-ray crystallography, you're a little bit locked into one particular state of a protein. But as you know, enzymes move around, they're dynamic, and so protein NMR is another method. So you don't need to know details about protein structure, about X-ray crystallography or NMR, but these are two of the most common methods used to determine the three-dimensional structure of proteins, and thus they are very important methods in biochemistry.
All right, so there's a few take-home points I'd like to emphasize here at the end of this lecture. So the first is about the proteome. So you need to understand that the proteome describes the protein content in a cell or a group of cells at a specific time under specific conditions. So while the genome is something that's fixed, the proteome is dynamic, so it can change in different cell types or under different experimental or physiological conditions. You also need to understand the general principles of protein purification. So you need to know that proteins can be purified based on things like size, charge, and affinity for other molecules. And also that once your protein is purified, it can be visualized using SDS page and isoelectric focusing. You'll also need to understand how antibodies are used by biochemists as tools in the laboratory to both identify specific proteins and also to isolate proteins from other proteins. So you'll need to understand ELISAs, Western blotting, and immunoprecipitation. You should also be familiar with how the primary sequence of a protein can be determined using either Edman degradation or mass spec. And lastly, you need to know that the three-dimensional structure of proteins can be determined using methods like X-ray crystallography and protein NMR. So I hope you learned a bit about methods used by biochemists to analyze proteins. And if you have a question about these methods or any additional methods that you may be curious about, please let me know.